let me get this. Okay. It'll turn on. Any questions while I'm getting logged in on this, guys? Have you guys, anybody started looking at studying for the lab stuff yet? I know a lot of people want to like deal with one thing at a time, but don't forget that you have the lab exam coming up too. And lab exam is after, you know, the break, and that's fine. That gives you some time to study over the break, but don't forget to at least like maybe look at what you need to study so you can get it organized before you get to it, so you know what to do. I would highly recommend that. Yeah. No, it's going to be in the lab. Yeah. So what I have for the lab, I have study guides. So those will just be like questions that'll kind of go over the topics and get you guys working with the information in a different way than what we have been. There's two different versions. There's one that's like more question based. There's one that's like draw this diagram or, you know, do things. So there's those. And then there's like a prep guide. And it says, I think it's called prep guide or whatever, but it's meant to show you guys what you need to be studying for the lab, because that's in the lab documents area on modules. Like you go to modules at the bottom, there's like all the lab stuff. And so it'll be in that area. But it just is going to show you, and it's going to show you what pretty much exactly what I'm going to tell you now, which is just that the lab tests, and like I've said it before, but it really, I'm not kidding about it. It tests over the stuff on the front of your sheets that you all have been writing on the back of. That's what it tests on. Everything, like I said, from day one that you guys need to know for the lab is on the front of those sheets. And there's not going to be a study sheet for those because those have already watered it down considerably compared to what's in the lab manual. So that's what you guys need to know for that. So there's the content part, and then there's like the steps for the procedures. Know the content part for sure. I recommend that to everybody. Like that's the stuff you will be tested over for sure. The stuff in the procedure, I'm not going to ask you how many minutes or how many milliliters or whatever that is, right? I want you to know concepts for it, but look through the procedure and make sure it makes sense. Because if there's something you look through in the procedure and you're like, oh wait, but why did we do this step? You might want to know why you did that step. So stuff like that. It should outline it in the top, but maybe you missed it. And so just look through the procedure and make sure that you get it. Yeah, yes, yes. So I have, and that's okay. If we're going to be, because I know it's getting close now, everybody's like actually like real time thinking about it. But yes, so it's going to be at the benches, and like each seat where you guys sit at in the lab will have like 2 to 3 questions at it, and we will rotate from station to station, 1 minute at each station. So it'll be like 1 minute, and then you go to the next 1 minute, and you go to the next. There'll be microscopes there that you will look into. You don't have to focus them or operate them. I don't want to deal with that right now. We might look at that in the final if I get frustrated enough about a microscope situation. I just didn't have the energy to deal with it while I was sick. So that's that. But yes, so you go from that, and there'll be microscopes that are already set up, or there'll be like a picture of something, or there'll just be questions sometimes. It just depends on, you know, whatever it is. 2 to 3 questions at each place. I say there's a minute per station. There's going to be 25-ish stations. Sometimes there's less, and there's 24 to 26, you know. But anyways, and they'll be numbered, and I'll help you guys walk through them and whatever. After you've gone through each station once, so it's 25 to 26 minutes, you know, everybody can go back to whatever station they want to and revisit it for as long as you need to. But I just want everybody to get at least, you know, the same amount of exposure, chance to look at every question before, you know, they get blocked out and don't, you know, whatever, by everybody else. Yes. It is multiple choice. Yes. For now, for everybody, for now, as long as I feel comfortable with it, I feel like multiple choice is sufficient enough for what we need to know for this course. Maybe people in the future will be unlucky and have to have, like, you know, write the term or whatever. So, yeah. Yeah, and I don't want to have to like interpret people's drawings and like they drew it right, but I just interpret it wrong. I don't like putting that on people's plates, especially whenever you guys aren't meant to be experts on these topics. I just want to make sure you understand them. You know, I want to make sure you get the content. So yeah, no, there will be multiple choice. It'll only be up to 5 choices. Scantron. So whatever you have on Scantron available. So I will provide the Scantrons. Please bring your own pencils and erasers if you need to. Any other questions about it? Yes. Yes. So I did it. I typed it. I know I typed it right. Okay, state.edu. There we go. Yes, so I'm going to do, we'll have reviews coming up. If you guys look in that thing that I sent out of like all these dates of things coming up, you can go back and revisit that. This is like one of those times in the semester where you might have to go revisit that announcement a lot, because it's like we'll get one thing out of the way and then like 2 days later, there's another thing coming up, and like 2 days later, whatever. A lot of them are reviews, so not necessarily necessary, or you might just want to watch them whenever they get recorded, and that's fine too. But keep a lookout on those. We will have a review for the actual, so we'll have the weekly review. You guys know how we do that, usually of whatever we learned. We have our weekly review for chapters 13 and 14 coming up on this Friday. We'll have it at the same time, 10 a.m., usual thing. Then on Sunday at 4 p.m., I figured 4 p.m. was a pretty decent time just because it's not too late. You can still have time for dinner and whatever. We're going to have at 4 p.m. on this Sunday, have a review for the Unit 2 exam. And so that's mostly going to be playing the games like we did for the Unit 1, just chilling, and you guys can play through the games. And then I figure after we do an hour of playing the games, then I will, what I'm thinking of doing, because we haven't had during Unit 2 much time to go over the in-class questions. And I know some of that, like, we're glad that we don't have to get called out, and that's great, but maybe we would like to learn that material we could have gone over, right? So what I'm going to do is after we do the games, I will have already posted somewhere, I haven't done it yet, but when I get the chance to, all of those questions that we've missed, that we haven't gone, all of them, just say, let's all of them from Unit 2, okay? And we will go over all of them after the questions. So if you don't want to stick around for that, you don't have to. If you just want to watch a recording of it after, you can, whatever, but we'll do the quest, we'll do all the games, and then we'll do the in-class questions after that, review that, so that there's a little bit more interaction involved there and whatever. And I don't mind doing that. It'll take probably a little longer, but that's okay. I know people would rather, you know, if it's recorded, you can go watch it on your own time, or if you have the time and you don't want to miss out on it and it helps your test grade, then of course you want to participate. So that's an option as well. So that's what we'll be doing for the lecture. And that'll be on Sunday, like I said. For the lab, and of course we have, after our lecture exams, we have fall break. So that's nice. So we can just knock out that test, and then like we have time to chill and slowly, generally not crazily focus on the lab stuff, hopefully. The lab, we will have a review. It's in class. It says on the syllabus that it is optional. You are welcome to, I'm going to have it on Zoom in the lab. It's like if I was doing Zoom, but in the lab, okay? So if you wanted to do that at home, you can. If you wanted to watch the recording of it later, you can. Or if you want to be in class and actually participate in it, you can. I'm gonna be giving it as not a game, but as an actual, like we do with our questions here in class. I'm gonna walk through the questions, okay? And I will be probably in there being like, okay, so that's what this question's about, but remember, you also have to know blah, 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 and remember how that works, because blah, 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 blah. That stuff's gonna happen during that review. So you aren't doing anything else probably during lab time, so I highly recommend that if you can, during your regular old lab time, just sit there and play this game and listen to me blab like you would during that review. It won't even take the whole lab time probably. Come and sit and just chill and take notes with me in person, because that way you're there in person and can ask me questions there. And it should make it a little bit easier. That's the review that we'll be having for the lab, and that's the only review I will be having for the lab. There will not be another one like at night, the night before or something. So that's the review. And it is Kahoot based, but again, it's going to be a lot like we do in the mornings here, where we have questions and I kind of walk through it with you. It's going to have a little bit more to it, but yeah. And it's kind of helpful because it kind of sets up, because even though I've been saying, yeah, you guys have to know all this info on the front of the sheet, and it's hard for me to like pinpoint kind of what that means, but the questions put it into perspective. So it'll be something like, okay, what is the primary stain for the gram stain? Or what is the primary stain for the endospore stain? Which of the following stain procedures used heat? and stuff like that. So you can actually think through that stuff and apply it. I recommend coming to that review somewhat prepared. I don't mean you have to be ready for an exam before the review, but like at least look through your notes so that some of the old stuff is, you know, you remember it and you can actually engage in it and apply it a little bit when we do the review. So you can get something useful out of it. That makes sense for everybody? So that's what's coming up, all right? Thank God there's a break in the middle of that. All right, library. So we're gonna do, this is 10, chapter 10. I have a hard time finding what I'm trying to do on this. All right, let's do it. Oh, let me pass around the sign-in sheet too, I forgot. Oh, and I didn't ever set this. Yes. All right, let's do it. All right, which term refers to all of the chemical reactions that occur in the cell? All right, so the term for that is going to be metabolism. Metabolism is overall all the chemical reactions in the cell. And then the way we see it in like macro creatures, we would see that as like the whole chemical reactions that go in the whole organism. So yeah, this is annoying. Alrighty, next question, anabolism. What is that? Alrighty. So anabolism is the building of things. Remember, anabolism and catabolism. Cats tear things apart. Anabolism, like anabolic steroids, we're building up muscle, so we're building things. We need energy to do it. All right, enzymes are an example of what macromolecule? Why is it so fast on this one? Some are so fast and some are short. And I wrote all of these by myself, so I don't understand that. But whatever. So yeah, enzymes are proteins. For the most part, for our purposes, enzymes are always going to be proteins. Amino acid sequences, all curled up, right shape, doing work. Where does the substrate bind to? right? So this is called the active site. That's where the substrate that the enzyme is actually acting on binds to. So pretty straightforward one. What are the causes of a denatured protein? How can you denature a protein? Sorry, I know we have the crappy blue pen. Someone smuggled away my good black marker, so they switched it out with their crappy blue one in lab, I guess. All right, so pH, heat, and chemicals. This is exactly right. So definitely pH and heat is one we've talked about the most. Chemicals are an option as well. I think we've like grazed over that one a little bit whenever we were talking about it, but we're going to get more familiar with those in chapter 11. But they can operate in a similar fashion to the pH and the heat. All right, what best describes non-competitive inhibition? We're inhibiting non-competitively. Okay, so non-competitive inhibition refers to attaching to the allosteric site. So what this means is basically it's an inhibitor that, so allosteric is another word for regulatory site. And it's a term that is present kind of in, through the text and through some of the present, like the lecture notes as well. But it's the same thing as the regulatory site. So somewhere else on the protein, we're binding with usually a product that has built up and then causing a change in the enzyme shape so it can no longer function the way that it is supposed to do. So that's why yellow isn't the right answer, because an inhibitor that allows normal enzyme function, well, if an inhibitor allows normal enzyme function, that's not inhibiting it, is it? So that's also a problem there. So it's going to be the red. Allosteric, same thing as regulatory site. The 5-carbon sugar found in ATP. Everybody's always familiar with the fact that ATP has the 3 phosphates pushing against each other. What are the other 2 components? 5-carbon sugar. And that 5-carbon sugar is going to be ribose. It's just a nucleotide, like a ribonucleotide with extra phosphates. Right, aerobic respiration requires what? All right, so most of you guys got this one, but I made the choice pretty easy here. So obviously aerobic respiration is going to require oxygen. And this is a concept that I tried to drive home quite a lot because for some reason this can be a commonly missed question on the exam. We kind of touched on that in the review over the weekend, but oxygen. So aerobic, there's only 3 ways you can make energy, right? There's going to be aerobic, anaerobic, fermentation. Only those 3. There's no others, right? So if you can remember that fermentation is the short path where we're only doing glycolysis, that's it, right? And then we're dealing with byproducts made to, basically deal with that stumped off situation. Aerobic and anaerobic, both of them deal with the whole thing. Both of them deal with the whole thing, okay? So they both have glycolysis, they both have Krebs, and they both have the electron transport chain. When we go through the electron transport chain with aerobic respiration, when we get to the end, we need a final electron acceptor. And for us, that was oxygen, right? And it made water as our byproduct. We split the oxygen, added on the hydrogens with the electrons, and that's how we dealt with those protons and the electrons that were building up from the whole process. Our final electron acceptor, therefore, is oxygen. However, when you're talking about anaerobic respiration also goes through glycolysis and Krebs and the electron transport chain, but it just doesn't have oxygen. It uses something else. That's the only difference. The only difference there that you have to worry about. Okay, so there's 3 ways. And I know this, everybody got this right, but it's commonly missed on the exam. So just don't forget about that. It seems easy now. Aero, air, oxygen. I say like air, AIRO, oxygen. That's what I try to like drill it into people's heads. But when I ask you what the final electron acceptor is of aerobic respiration, you guys do need to know that. The gas produced by yeast in alcoholic fermentation, what would that be? Even things like this, if you guys can at least try to remember, like, you know, alcoholic fermentation. Okay, what do we care for that? Okay, alcohol, we're making alcohol. When I drink alcohol, what kind of gas is there? It's going to be CO2. Okay, that's the gas that's there. Is ethyl alcohol there in alcoholic fermentation? Of course. Is that a gas? No, that's a liquid. Last time I checked. Last time I checked. But yes, so don't get caught up in that stuff either. Don't forget that it says gas. If you see alcoholic fermentation, the answer is not just alcohol. Read the whole question. All right. During photosynthesis, plants use what gas to produce glucose as their food? This is a long one. Where is my phone? Oh, it's recording. I'm like, what do I do with my phone? It's right there. All right, so this one is carbon dioxide. Exactly, we're going to take that inorganic carbon source and fix it into an organic one. And then we can use that to build the plant parts and make energy in aerobic respiration. Good job, guys. Let's just put that down there. Maybe that'll be back later. Okay, so we pull up chapter 13. We'll do 13 and 14 today. Yeah, we're gonna do it. I promise we'll get through it. Okay. So chapter 13. Chapter 13 is dealing with disease and presentation of disease and how we're defining that. And basically how the body can be susceptible to disease. Disease being, of course, deviation from health at all. Any deviation from health is considered disease. So first of all, we know we have a microbiome on our bodies. You don't just have bacteria that naturally live in your gut. We also have them that naturally live in other areas of your bodies, like your skin and your mouth, maybe on your scalp, wherever, and, in the reproductive tract. All of these areas can have a normal bacterial flora that we expect to be there. When we are talking about you plus your flora, that term is holobiont. So again, holo being the whole, like it was with the enzymes, right? So including our biota. Okay, so anytime we're talking about disease, again, that's any deviation from health. It does not necessarily depend on infection or anything like that. No pathogens have to be involved for disease. What I mean by that is if you guys have ever heard of diabetes, that's a disease. Cancer, disease, these don't depend on infections, right? So don't forget about those guys. Things like, heart disease and stuff. Those are still disease. They're just not caused by infection. Of course, in this class, we're focusing on the ones that are caused by infection. And an infection is a condition where we have microorganisms penetrating our defenses, entering the tissues, and multiplying. All right. You might have E. coli living in your colon right now, just doing normal E. coli things, as long as it's not the bad version of it. And it's just chilling. And I promise that you probably do have E. coli in your colon. And it's fine. That's not an infection because it's doing what it's supposed to do, where it's supposed to be. Nothing weird is happening, right? If it's actually penetrating the defenses, entering a new area of tissue, overgrowing where it shouldn't be overgrowing, things like that, that's where it's going to become an infection. All right, your normal biota, really all you need to know about what your normal biota can consist of: bacteria, fungi, protozoa, and viruses. There's 4. There's a 5th group that isn't included in here. That's the helminths. The helminths are not part of your biome. Pretty much everything else in that categories anyways could be part of your biome, potentially. So that's all this is talking about, regular stuff you can find regularly in your body. I know I'm aware of what TikTok will tell you about a helmet in your body. Okay. I'm aware of what TikTok says about clearing out the worm infections that you have that your doctors are lying to you about. Okay. I'm aware of it. These are lies. I promise. Go away, and then you'll clear out all the worms in your body, and you'll see them in the toilet. It's like all this stuff like this, and like you'll see it too. They'll like take pictures of the toilet, and they'll have like they'll say worms in it. 99% of the time, what's happening whenever they take these weird supplements, or they do like the lemon cayenne drinking BS, or they do the they take ivermectin sometimes. That's like over-the-counter ivermectin that's meant for like animals typically to try to clear out their worm infections. And then they'll show like, oh, okay, but we cleared out the worms. See how we weren't lying? That's your intestinal lining that you're sloughing off. Congratulations. Like that's what it is typically. Huh? Yes. Yeah. I don't know if like the people that are selling it, like trying, like I think they're trying to get views, but there are people that watch it that believe it, and that's what's dangerous. So anyways, that's not real. In case you guys didn't know, you don't have worms in your body. I'm sure any GI specialist that does colonoscopies, like hundreds of them a day, can tell you that there's not. Okay, so yeah. If it was so easy just to clear out all the worms in your body just with those, you know, prep things that they make you take before a colonoscopy, everybody would just be doing that in third world countries. So, but they're not. It's not that easy. All right, so I would believe if they were there, we would know. Anyways, normal, and also there's this whole other thing we're going to get into later on with the unit 3 that we'll talk about with immunity. It's called the hygiene hypothesis. If you guys haven't heard about it, it'll be really interesting, I think, for some people to learn about why you have allergies here in the United States and why they don't have allergies at all in third world countries. So, interesting, but there's a reason for it. Anyways, normal biota can be anything except for a helmet, basically. That's what this is getting at. And there will probably be a question about it on the test of like, which of the following can be, and there'll be 5 choices, and you have to like, or can't be, and then you have to choose that can't be would be helmet or something like that. All right, looking at what you're colonized with, what's causing infection, and then how we're being clearing it out or whatever. There are different ways that you can be colonized. I know that this is not actually plugged in. I'm imagining that it's plugged in. Yeah, it's not actually plugged in. There we go. So resident microbes, things like the microbes that are in your gut, like the E. coli, whatever. A few potential pathogens, things that can come into your body, let's say like COVID or like, you know, salmonella or anything that could be causing potential infections, right? And then we have transient microbes. And these are things that you're exposed to all the time that you're just clearing out. So like you go outside and, you know, if you were to drop your lollipop on the ground and eat it, but you didn't get sick or anything, right? Because that probably happens. Kids do it all the time and whatever. Or like, I don't even know what else. There's all sorts of stuff. Like you set your at your own home, set your piece of food on your countertop. That countertop's probably not sterile, right? It's probably got some bacteria on it, and it gets on your food, and you eat it, and you don't get sick. Why? Because your body's just clearing it out. That's transient. It's just making its way through, or it gets killed. That's usually the more likely scenario. So your body's getting rid of it. It's transient. You never knew it was there. Nobody cares. So that's the transient guys. All right, so we have our resident microbes. There are green guys, let's say good guys that are kind of taking up, I like to say my analogy is taking up the stadium seats. So if you guys are at a big old stadium, there's all these seats available, and as long as you have a healthy biome going on, the healthy guys sitting in all the stadium seats. So there's not space for the bad guys to sit down. So should be good, should be fine, right? And then our potential pathogens, they have to find a place to be somewhere in your body by latching onto tissues that aren't colonized normally or moving into tissues that are deeper where there shouldn't be any microbes and things like that. So a lot of things can come along and just get exit from the body. So if we say potential pathogens, just because you're exposed to salmonella doesn't mean that you're going to get salmonella. So your body could just like, it could be transient, pass through, act as though it is transient and just be cleared out. You never know that it was there. However, sometimes pathogens like salmonella, and sometimes even our own resident microbiome can cause infection. So you have your microbiome that's supposed to be there on your body, like the normal bacteria on your skin, like the Staphylococcus epidermidis and things like this. What happens we can be, for example, what was currently recently just my issue that you guys were all made aware of because I can't keep my mouth shut about anything, which is that urinary tract infection, right? 80% of urinary tract infections are going to be caused by E. coli from your own body, your own bum, basically, right? And it's usually either going to be from if you have poor hygiene practices when you're using the restroom, and women are always told to wipe from front to back for that reason. This is the reason why. Or if you're having sexual activity and there's just a lot going on down there, that's why we are recommended after sexual activity, you're supposed to urinate and maybe wash up or whatever to help prevent those bacteria from taking hold in your urethra. Because if you get normal E. coli that is part of your resident microbes, the green guys, they're supposed to be in your body, but they're not supposed to be in your urethra or in your urinary tract, right? So that's where infection can instill itself with your normal microbiome. If it's going somewhere where it shouldn't be. Another example of what can happen with your microbiome causing disease. If you have, let's say, a healthy microbiome, all your stadium seats are taken up by a grand variety of things like E. coli and Klebsiella and Enterobacter, and even maybe a little bit here and there like C. diff or something like that in your gut. There's a ton of bacteria in your gut. Now let's say you go to the hospital and you have surgery and you're on antibiotics, and it clears out the entire stadium except for C. diff because it's resistant. It's got the endospores. It can just endospore down. It doesn't care about your antibiotics. And now you've got a problem because C. diff is sitting there like looking around being like, oh, look at this. I got all this space. And then it multiplies and takes over the whole stadium. And that's where you get infection as well. It didn't ever leave where it was supposed to be. It just overgrew, right? So we know that's an issue. Another one that we would probably be common with, because a lot of this stuff happens all the time. Another one you're common with would be things like yeast infection. So you take your antibiotic for whatever it is, even your UTI, and you clear out your normal bacteria in your flora and like the vaginal area, and then clear out those bacteria, and now yeast can overgrow. Yeast are normal. They should be there, taking up some of the stadium seeds. But if we kill off the bacteria, now they can overgrow and cause a yeast infection. So it creates this sort of vicious cycle of now you treat the yeast infection, but do the good bacteria grow or do bad bacteria grow and cause BV, which is bacterial vaginosis? All of that is a problem if it's not being handled appropriately. So BV, it depends, right? So if you have resistant bacteria that are resistant to the antibiotic that you are taking, and it kills off the healthy bacteria that would have been in your normal flora, and the bad ones, I don't say bad, but the balance gets thrown off. Yeah, the balance gets thrown off because it's usually, normal bacteria shouldn't be certain levels of certain kinds, right? That can happen from UTI or more commonly from taking, like if you have a yeast infection and taking medication for the yeast infection, now you've cleared out the yeast stadium seats, but the other bacteria can grow up faster maybe than the healthier bacteria. So it's just a matter of what antibiotics you're taking for what and what antifungals or whatever it is. There's a lot of different ways that you can get either one. Additionally, if you have thrown off your flora, let's say like your normal population of what's in your stadium seats, and it gets messed up from sexual activity or new body wash or not controlling the pH or I don't know you started running and you run in the same shorts every day and you're not keeping up with stuff like that or whatever it is can throw off the flora in that region pretty easily, and that can lead to BV as well. So these are common things, but they are often associated with normal biota. We didn't have to introduce anything new from your body. It's your own body doing this to you. So that's kind of interesting to think about. We always think about the pathogens that are just pathogens. Things like salmonella, that should not ever be in your body. Things like, the plague, that should never be in your body, or COVID, right? Anyways, obviously we know what disease would be, could lead to disease. Infection, you can have infection, and infection is a separate thing from disease, right? So infection, overgrowing in a population, deep in tissues, things like that. Now we have growth of this bacterial population where it shouldn't be. That can happen without any symptoms. So if there's no symptoms, then it's not really a disease because that's not a deviation from health if you didn't know that it was there or not, right? When you have the symptoms or the signs associated with it, and we start deviating from the health, actually deviating from it, that's disease. So they don't always, they're not one and the same, is what I'm saying. They're not the same thing. All right. Human microbiome project is in place to try to figure out what our normal biome should be or could be compared to certain groups of populations and things like that. We have this special situation called microbial antagonism. This is a term that you need to know for this chapter. Microbial antagonism is when all of your normal good guys in the population that they're supposed to be are taking up your stadium seats so that bad guys can't get in. So they are serving a purpose there, basically having a, blocking the bad guys from sitting down, or they could be making byproducts, keeping the bad guys away, or using all the food in the area so the bad guys can't take hold. Whatever it is, when your normal microbiome is helping defend against infection, that's microbial antagonism. All right. There's a lot of factors involved in getting an infection. I'm not going to go over this. If you guys want to look at it, you can. I feel like these are things that are kind of obvious. So we can look at exogenous and endogenous infections. So endogenous infections are infections caused by, microbes that are already there. So like we already said, like with E. coli causing your UTI, with C. diff overgrowing, or in this case, we have Streptococcus pneumoniae. This is a very common bacteria, gram-positive bacteria. It's carried as your normal biota. A lot of people carry it in like their sinus cavities, their upper respiratory area. And this can become pneumococcal pneumonia. It says in AIDS patients, if that is in your own biota, that would be the case. But you can pass on different strains of this guy to one another and pass on pneumococcal pneumonia, as well as this is the same bacteria that causes pneumococcal meningitis. This is the #1 cause of pneumonia and meningitis in the world. I'm gonna close this door real quick. Dr. Singh and I went to this talk or whatever. We have to do it every year, but it's like about active shooter situations. And now it's like we left the talk and we're like, have you been closing your doors? And I'm like, I don't know, maybe I should close my doors more. And now we're talking about like how often closing your doors or locking it or all these things. Yeah, I agree with that. So you guys should be fine. It'll be good news, everybody. But yeah, yes, you're right. I agree. That's probably true. But it's one of those things where we have to be trained up on it, and all that makes sense. But it gets you thinking about it. Yeah. All right. So where do you get your microbiome that's going to take up those stadium seats and keep you protected and digest your food for you and all these wonderful things that they do? We believe it starts off in the uterus, just being in the womb, which should have a little bit of its own biome. The womb itself doesn't have a lot of a biome in the uterus, just in general, even when you're not pregnant. It doesn't have a ton of a biome, but we do think that that's where it probably starts. Science says, like really, really probably does. The most, however, the most exposure that you're going to get is through birth. And this is coming through the birth canal or being born by C-section. And you can imagine that this would be a very vast difference from one another. If we're having somebody being born through C-section and we have, sanitized that area, disinfected that area as much as we can, almost sterilized it if possible, and then pulled somebody out into that environment versus pushing somebody through somebody's vagina, which is definitely not sterile, you're getting exposed to really different bacteria as your initial exposure to new bacteria. And this can be both a good thing and a bad thing. It's good if you're being born through a birth canal that has normal flora and should be relatively close to what it should be. If you have gonorrhea, if you have herpes, if you have something like this, or some certain kinds of strep, then being exposed to those bacteria could be deadly to the baby. So it's a give and a take, right? So it should, otherwise, healthy people should be a good thing, giving your baby a new exposure to bacteria. Milk, breast milk, that's going to has its own microbiome on its own, and that is a great way for babies to be exposed to bacteria that can digest sugars that they cannot digest otherwise. Most sterilized formulas aren't going to have that. And so that could potentially lead to digestive issues down the road because that baby didn't end up having the ability to break down milk and certain things like that appropriately. There are formulas, of course, available now that we're aware of this problem. So now we have formulas that are probiotic. So yeah, or have been recalled. Like you had those other bacteria, right? So this is a depends on how you're going to look at it. But Yeah, so you can buy them with probiotics to help encourage this biome developing. After this, we have encounters with caregivers and other people, and then the environment, of course, your kid crawling around on the ground, a dog licking them on the face, and all these things, picking up bacteria that way. Now, once you get your biome as a kid and you've taken up all your stadium seats like that, are you done? Is your biome like that forever? No, right? We know that it's not. When you go, especially, it could change throughout lifetime, of course, through any exposures to different things, but you go through puberty, that's a big change oftentimes because of your pH and your body's going to change, the hormones and what's being regulated and how, so you can get a major shift in your biome then. When you become sexually active and you're being exposed to other people's biomes, and it can be incorporated within your own or throwing off your own. And then as you're aging and your immune system is changing as well, I mean, it's going to be changing pretty much constantly is what I'm saying. So it's not set in stone, but that's how we think it gets started. So how does infection progress? We start off with a pathogen. This is a microbe with a parasitic relationship with a host. Okay, so a microbe that does this and can lead to infection. Pathogens lead to infection and can lead to disease. True pathogens are capable of causing disease in healthy people. These are important. This is an important one to be aware of. True pathogens cause disease in healthy people. Opportunistic pathogens cause disease whenever the host defenses are compromised or when they're somewhere that they shouldn't be. Not natural, sure. So opportunistic pathogens. You normally have E. coli, it's not causing a problem, it gets into your UTI, it gets into your urethra and causes a UTI. That's the bottom one, right? Establishing in the part of the body that's not natural. We're talking about host defenses being compromised. Opportunistic pathogens. Here we're talking about a shift in the biome from taking an antibiotic and throwing off the levels of bacteria in that area, for example. So that would throw off our host defenses. Because part of your defense is your normal microbiome. Okay. See the difference, yes? Virulence is a term referring to the degree of pathogenicity. So pathogenicity, we've already talked about pathogen, this is a disease-causing microbe, right? This is pathogenicity, like basically talking about how it can cause disease. The degree of its ability, though. So now the degree now, how bad is it? That's what virulence is talking about. How bad is this disease that we're causing going to be? So this is usually indicated by our ability to establish infection and cause damage in our host. And anything that allows a microbe to do those things, establish infection and or cause damage in a host, we call those virulence factors. Makes sense. That's a common term that's going to come up over and over again in the course. Will not be going away anytime soon. I know I've already said it a couple times before. So anything that helps with that, like flagella, like a capsule, anything that helps with that. All right, the infectious dose is the minimum amount of microbes you need for an infection to be established. That's all it is. With a virus, it could just be one virus. With like tuberculosis, for example, you need 10 tuberculosis bacteria, typically on average, to cause an infection. That's pretty low for bacteria when we think about salmonella being like 50 to 60 bacteria needed to cause the infection. So like I said, some of them can be pathogens, but be transient. If you only have like 5 bacteria, salmonella get into your body, it's probably not going to cause an infection. Get kicked out before it can. And so here's how the steps that are involved in microbes causing disease. So we're going to go over these briefly because I feel like a lot of these are kind of straightforward. Finding a way in. You can get in through broken skin, the GI tract, eating, breathing them in. If you're exposed to like somebody sneezing, for example, the urogenital tract, maybe from STIs, and then endogenous biota that's normally already there. So that's your entryway. Attachments and negotiating the biome. For bacteria, fimbriae are little extensions that are like hair-like extensions. We learned about these, or attachment. That's what they're for. They also can have capsules that makes them sticky. It also helps them evade the host defenses. We have surface proteins on some bacteria and or viruses, which we would call viral spikes if they were on viruses. Those proteins interact with certain kinds of receptors, like let's say certain proteins on liver cells, like we had mentioned before, or whatever it is. That's their way of getting into the host. Surviving the host defenses, you have to avoid phagocytosis or avoid death inside of the phagocyte. So you're going to get eaten up by a white blood cell at some point. how are you going to maintain your infection levels, right? So, or you could have an absence of very specific immunity. That means you're a microbe that basically evades the antibody response. Then we have how you can cause damage, which is going to lead to presentation of disease. So this could be from toxins that you are secreting. Those would be exotoxins. Endotoxin, remember, there's only one thing that has endotoxin, that's gram negatives. All gram negatives have it, but endotoxin is only endotoxin. LPS, lipopolysaccharide, endotoxin, but it is a toxin, and it does lead to inflammation and cause septic shock and things like that. So we can have toxins and stuff that are associated with this that can trigger this damage response. We can also have an excessive host response. So causing the host's immune system to be triggered far more than it should be can cause damage. That can be, I would say, on purpose. It could be in favor of the bacteria or virus, or it could just be something that just happens to happen and isn't really good for either party. We can also cause epigenetic changes in the host chromosome. That just means changing how our genes are expressed. Things like, HIV inserting itself into your genome can do stuff like that. Okay, you can change how your genes are expressed. Then you have to get out of your host once you have grown a lot and you want to move on to the next guy. These are pretty self-explanatory as well. Respiratory, salivary, skin cells, fecal matter, urogenital tract, urine itself, and then blood. All right, so we went through these. I'm not going to go through these in any more detail than what we did, but definitely know the difference between exogenous and endogenous infections. Exogenous, it came from outside of you. Endogenous came from your existing flora. Yes. Yeah. Right. So it's not, so it's easy to say that whenever you and I are leading lives like we are here. So go ask the heroin addicts that question, because they probably don't agree with you. They would say it's actually really easy to get hepatitis C or HIV because they're sharing needles with people. So these things exist, right? And they're pretty prevalent in our society. Did you know you can bleach your needles? And it's don't do it. I'm not recommending it. Or like, if you guys are going to be working in ERs and you have, people who are, intravenous drug users and they're at risk for a situation like that, if they're going to have to use it, then I recommend that they, bleach their needles before they share them. But yeah, you can bleach them and just like rinse them off and then go on with your heroin addict problems and do your life that way. This actually was in the notes, in the slides, and I saw that one time. It was like a couple years ago. I noticed it the first time, but it's nuts. But yes, so bloodborne infections, there's that situation where we're talking about intravenous drug use. There's also, of course, we can talk about arthropods. So arthropods are insects that are like biting, they drink your blood, and then they can pass it on to other people, which is a huge issue with things like malaria. It's also how things like, West Nile and stuff are going to be transmitted. So in the blood that mosquito drank up. Mosquitoes are really heavily disease-carrying insects, very much so. They carry thousands of diseases. I can't even begin to tell you. It's ridiculous. And a lot here in the US. Anyways, moving on. We know that the whole issue with the baby can be a barrier with placenta, can be a barrier that protects the baby, but it can also be a means of, the baby getting stuff from the mother. Just depends on the disease, if it can cross the placenta or if it can be maintained within the mother. If you are a mother who has certain kinds of diseases that you can keep relatively in check, but you're already like, let's say HIV positive, you have HIV, you take your medications, your baby can be born like high incidence of being HIV free if you take your medications. It's something like 96%, if not more than that, as long as you're on your medications. That's pretty impressive. So we can keep the virus from getting to it that way, but not all things can be prevented that way. And some things just won't cross the barrier and the baby will be fine. Some things cross the barrier and it's worse for the baby than it is for the mother. Things like listeria, for example. Anyways, we talked about adhesion and how you're going to stick to things. In the past, we've talked about quorum sensing. This is how organisms are going to basically communicate with one another from waste products and reactions to like nutrients in the area. They can kind of read that around them. But this is the pictures of what I was talking about, the attachment mechanisms. Phagocytes are our white blood cells that are going to eat up our bad guys, right? Some of our bad guys, our bacteria, viruses, whatever it is, have anti-phagocytic factors that either help them evade phagocytosis or help them survive in a phagocyte, something like that, or help them kill a phagocyte. All right. So you can, like I said, kill, that will be a variance factor, anything that helps you with that, some sort of toxin that you're producing, for example, or you can evade. This could be making it difficult for engulfing. So this could be like what we said with our capsule. And then adapted to survive inside of phagocytes. This is what we see with tuberculosis, for example. We'll go over that more when we get to that chapter, whatever it is that we do, chapter 24 or something. 23, I don't know, 22. Virulence factors. We already kind of talked about these, anything that helps establish infection and disease. Oh, all right, how can we cause damage to our host? Usually excreting enzymes that can break down the cells and the barriers to help us cause infection. So again, these are going to be exoenzymes that are secreted outside. Exo are going to be secreted. Examples, hyaluronidase. This will be breaking down hyaluronic acid. That's part of your extracellular matrix. We already talked too about collagenase. If there's anything that's like an enzyme like that that can break down those barriers and allow them to get in, those exoenzymes are doing a favor to the bacteria to help them infect more easily. All right, a toxin is anything that can cause poisonous effect, so can be damaging to that cell. There's exo and there's endo. And like we said, exo is anything that's secreted. These are toxins that are secreted. And then we have endotoxin that is always going to be for gram negative, and it's always going to be LPS. It's always this. It's not secreted, it's endo, it's part of the cell. We talked about hemolysis previously, but remember enzymes are involved in breaking down these red blood cells in order to achieve hemolysis. We talked about gamma with not having any visible hemolysis, alpha having greening because we have partial hemolysis, and then beta being where we have complete clearing because we have actual true hemolysis going on, breakdown of those blood cells. That's done by enzymes that the bacteria produce. You see down there strep pyogenes. This is Streptococcus pyogenes. This is a very common bacteria that causes bacterial infections like strep throat, necrotizing fasciitis, and other things. Not usually. Not usually. I think it can in some people depending on how they react to them. I think it's just because of the coughing more than anything, or like just like the damage that actually is done in the lung itself. And the lung is a little bit more sensitive than the throat. I think that's why. It's an interesting question. Anyways, excessive host response is something that can benefit or harm that pathogen's relationship with you, but it just can happen in some cases. Some organisms have adapted to it for it to be favorable. What I mean is, if you're having an overactive inflammatory response and you're causing a lot of damage to your own tissues, this can help some pathogens spread to those tissues while they're damaged and the barrier is down. So that can be helpful. Sometimes it just happens as part of the infection, like with COVID-19. If you have COVID and you get long COVID, it means that you probably had an overstimulated immune response while you were, actively infected. It's usually not infected for a long time, it's just symptoms lasting a long time. There are different patterns of infection that can be established. So we have localized, this is sort of self-explanatory, like a boil, something like that's just in that one area, or systemic. This is all over in your body, like sepsis, okay? Next one is the focal infection. This would be having an infection in one area of your body and it getting transmitted to another part of your body. So if you had like a horrible sinus infection that led to endocarditis and that spread the bacteria to your heart, for example. It's the same bacteria, it's the same infection, but it's not all over your body, but it's not local either. So that's what focal would be. Mixed infection, you have different kinds of microbes involved. A lot of times this will be the case when you have upper respiratory infections. And then you can have secondary and primary. Primary, let's say you got your UTI, and then as a result of you having the UTI or any medication you took along with it, now you can have a secondary vaginal infection like a yeast infection or something like that. Okay. For patterns, this talks about all that. I forgot that there were slides for these. Acute, we kind of know what these are. Acute is going to come on rapidly and have short-term effects. Chronic, it can come on slowly and progresses usually over a long period of time. Usually you have less severe effects in a chronic situation. All right, let's talk about the difference between signs and symptoms. All right, signs, symptoms. These are different things. They are not the term that you use. We often use, and I even would use symptom as a blanket term, but they're not the same, and you guys do need to know the difference of these. Signs, it says any objective evidence. Signs are measurable. Measurable and observable. Yeah, right. That's a sign, and you can definitely see that. So other signs that are obvious that you can measure, observe, things like that would be like fever, vomiting, right? Diarrhea. Can you see all of these? Yes. Can you palpate swollen lymph nodes? Yeah. So even swollen lymph nodes, red eyes, things like this that you can actually see. Those are signs. And then symptoms, these are subjective. They're reported typically. So what does that mean? Things like pain, you can't see or verify that it's there. Fatigue kind of falls under pain but headache. Nausea, you can't actually verify these, right? You can only go based off of what the patient is telling you about them. So this is hard, and or maybe infer, and like you'll have the advantage where if you're working with animals, they're not going to be lying when they're acting like they're in pain. So humans, however, will absolutely put on a show to get some, drugs. I bet you did, yeah. Some people, I went to the ER when I had pancreatitis last time. I went to the ER and I was sitting there for 3 hours to get in, and I was in that waiting area for 3 hours. But there's a lady out there in a wheelchair, and she was just sitting there. She's like, I've never been in so much pain. Why can't? And I'm just like, you know what she's there for. Like, you knew she was there to get pain medication. Anyways, it apparently happens like all the time. And of course it would, but apparently even more often than I would think it would. So yeah, you can't. You have to give them the pain medication, and then you can send them home with like a rehab pamphlet, but that's the best you can do. But you have to treat it. Once they're comfortable, you can pick them out, but that's it. So anyways, so these are our signs and our symptoms. Know the difference between the two. I always remember that a sign, if you could hold a stop sign, right? I know that's a different type of a thing, but if you can hold a stop sign, that's a physical thing you can interact with. There's no iteration I can give you of symptom really, where you could physically interact with that term in some way. So sign, you can interact with it. It's measurable, and that sort of thing. Symptom, it's just reported. Anyways, syndromes. So syndrome is a different thing than all of these things, but a syndrome is basically a disease that is defined by the signs and symptoms and not by what causes it. What I mean by that, we talked about meningitis, so it's a good example. So let's go with that one. Meningitis, light sensitivity, painful neck, fever, headache, and things like that. Maybe nausea, fatigue. Okay, so we could say that's meningitis, but what is the cause of meningitis? And it's not just one thing, right? Because it's not, right? We have several kinds of bacteria, a lot of different kinds of bacteria actually. There's viruses that can cause it, and there's fungi that can cause it, but it's all meningitis. So meningitis is a syndrome. Another one that you guys are very familiar with that you didn't realize that you're familiar with is the common cold. The common cold is not caused by one kind of virus. Common cold is caused by if not more, 200 different viruses can cause the common cold. So, but we all know the symptoms, right? The runny nose, you may or may not have a low-grade fever. You'll feel tired and you'll feel malaise and all of these things, maybe a little bit of soreness, but like it's a cold, we say, that's a syndrome. So we could have any sort of cause for that cold. All right, there's lists for examples for each of these for signs versus symptoms, and I'm not going to go through them, but they're good one to go back to if you are interested in reminding yourself how they work. All right, inflammation is usually the earliest symptom of disease. It's usually associated with the following. Edema, which is accumulation of fluid in a tissue that is being inflamed, and that is usually because cells that are sent there to do their job in fighting a bad guy are brought with fluid. That's why it's there. Um, Not, it depends on how you get it, right? Because you can have lymphedema, so it's just talking about fluid, lymph fluid being built up. And so it can be associated with hereditary problems. Usually, a lot of times we see that associated with things that we have poor circulation, like diabetes, and that could be, hereditary, or it can be, type 2 where you acquire it. Also, you see set up like that with like elephantiasis, where you have actual physical blocking of the lymphatic vessels by microbes, and so then you have buildup of fluid in those tissues as well. But yeah, it just depends on how it happened to you. All right, granulomas and abscesses, we're trying to wall off our infection. That's why they come out to be. And then lymphadenitis, anytime you see that, it's always going to be inflammation. So this is going to be swollen lymph nodes. I'm not going to go over all these terms, but I would be aware of them. Of leukocytosis versus penia, that's white blood cells being high or low. Bacteremia versus septicemia. Okay, do know this one for sure, because I will bring it up later, or it could be on the test. Bacteremia is any bacteria in the blood, even a few. I mean, there's a couple bacteria hanging out in your blood. They shouldn't be there. There's no need for bacteria to be in your bloodstream. It should be sterile. So if there's any bacteria present in the bloodstream, that's bacteremia. However, if they are flourishing and there's an active infection within your bloodstream, then that is septicemia. They are different. So of course, bacteremia could be a sign that septicemia is about to happen. But yeah. Viremia, viruses, fungemia, fungi. All right. We can have asymptomatic and subclinical infections, usually because the host isn't manifesting disease for whatever reason. Talk about those in a little bit, but then when you're done with your infection, you have to get out to move to the next place. We kind of already talked about this, but that's your portal of exit. You can have long-term effects from infection. This could be in the form of latency or sequelae. Latency is, we've mentioned this previously, but it's going to be whenever your illness is kind of dormant, right? You still have that microbe in your body somewhere, but it is not multiplying and causing disease. So chickenpox is the great example of it, where you're hiding that out either in your DNA, but also there are some with bacteria like tuberculosis, where it's just hanging out inside of your body and just not multiplying. It's just chilling. And that can happen for decades. But yeah, so any latency at all, virus or otherwise, it's going to be just hiding out. It's in your body still, but it's just not causing disease. All right, sequelae, these are long-term or permanent damage to organs. Like meningitis leading to being deaf, strep throat can lead to rheumatic heart disease, Lyme disease causing arthritis, and then polio can produce paralysis, pretty obvious one. So you don't have the disease anymore when you're paralyzed with polio, but you still have the effect of having had the disease, if that makes sense. It's called sequelae. All right, course of an infection matches pretty closely to what we would consider our bacterial growth curve. Our incubation period is, let's go to this thing. Our incubation period is whenever you are just no symptoms, the infection is just getting established, right? So when we were talking about our growth curve, this would be the lag phase. And this would just be getting set up, getting ready to grow. The immune system hasn't reacted yet, all that. That's your incubation period. For COVID, that can be like 14 days. Prodrome stage, the prodromal stage, or the prodrome, is whenever you have your first symptoms. This is whenever you get that scratchy throat and you're just like, I think I'm starting to feel bad. That's the prodrome. Okay. The acute phase happens during the height of infection. So all of this leading up to that, we would call this the log for the growth curve. So acute phase, you're having the worst of your symptoms, and it'll like at some point plateau out. You get worse and worse and worse, and then plateau out for your symptoms that you're seeing, signs and symptoms that you're seeing, maxing out. And then we have the convalescent period. So this up here where we've maxed out would be the stationary phase of our growth curve. And then the convalescent period is when you're getting better. Typically. Now, if you're chronic, you could go with a continuation period, but This convalescent period would equate with our death phase, right? So you can see how that lines up pretty well with how that growth curve will be moving along. And it makes sense as far as how we see our own symptoms when they develop with our diseases. Next, we have our reservoirs. This is the primary habitat in the world for that pathogen. Sometimes it's just humans, like we maintain the cold pretty well, but other times it can be, animals or soil or water or plants. Animals would be the carriers typically for like new flu strains, things like that. So those are the reservoirs, and then the source is distinct from that. This is going to be the thing where the infection was acquired from. So if you are going out and you get sick, the person that got you sick or the animal that got you sick, that's the source, but they weren't necessarily the reservoir, right? They could have gotten it from somewhere else. All right, our carrier is somebody who can spread a pathogen without knowing. So this could be asymptomatic carriers, so people who are just not showing any signs or symptoms. It could be incubating carriers. These are people who are infected but don't show symptoms yet. So those 14 days up to leading up to your first symptoms of COVID, you might be contagious as early as 5 days in. So you could be walking around for days not knowing that you're contagious and you had COVID. You didn't even know you had that. Convalescent carriers, these are people who have started feeling better and then went out into the world and started spreading the disease because they didn't get rid of it completely. Chronic carriers, people who are carrying it long term but aren't having serious symptoms. And then passive carriers, these are like medical situations. It's just an example, or dental people who are transferring from like one patient to another, not washing their hands or not changing gloves or something like that. All right, animals. Right, of course, we can transmit via vector or directly. We can actually also get sick from, ill people, what we usually think of, indirectly or directly. Directly is pretty straightforward. If I sneeze at you, if I cough at you, if I, kiss you or anything like that, that's direct transmission. Indirect transmission is like if you are, if I'm using this pen here and I was like licking all over it for some reason, and then I put it up here and then Dr. Rhodes came in and used the pen and got my COVID. From that, that's indirect, right? I didn't actually spread it directly to him, but he got sick from it nonetheless. So that's indirect. All right, you can have, okay, we were saying human carriers, a lot of diseases that you might not have presentation of symptoms or signs from. So those can be reservoirs sometimes. Arthropods are the most common ones that we would see associated with vector-borne. So this would be the pathogen being spread by something like a tick, flea, biting fly, mosquito, that sort of stuff. Arthropods is what they're called. We have biological or mechanical vectors. These guys are actually infected. When you get bitten by a bat that has rabies, that was your biological vector, right? He was infected with it. However, mechanical vectors are not infected. They're usually just spreading by contact. So what that means is the cockroach or the fly lands on a pile of poop and then comes over and gets on your food or on you or whatever it is, and then spreads the disease that way. So they were never infected. They're just doing what we did with the pen, right? They are the pen. A zoonosis is a term for any of these diseases that are maintained within animal populations. So there's a good table here explaining it, but of course, one obvious one would be rabies. Flu is usually maintained within chickens and birds, maybe pigs. There's a whole bunch. Rocky Mountain spotted fever. What do you mean? So that's the name of it, Rocky Mountain spotted fever. It's caused by rickettsia. It's like a rickettsial disease. It's bacterial disease, but it's just the tick-borne. And it can be deadly, actually. The spots can become hemorrhagic. All right, anyways, non-living reservoirs. I mean, the air, the soil, whatever. I'm not really going over those. Communicable diseases is anything that can be transmitted from person to person. There's no way we're going to get to the chapter 14 of this. I thought I could get it with you guys. I got it with the first day, right? Not with you guys. All right, occurs when infected hosts can't transmit the infectious agent at all. So that's communicable. Contagious is highly communicable. So it's a different thing. Contagious is highly communicable. So it's spread at all versus highly spread. And then non-communicable are diseases or infections, I should say, that can lead to disease that are transmitted, that cannot be transmitted from host to host. So if you fall and scrape your knee and it gets infected from the dirt on the ground, that's not really communicable, right? People aren't going to get that. All right, horizontal transmission would be like person to person. That could be the direct or the indirect, like we were talking about. This pen, when I explained what it was, it's called a fomite. And it is a type of vehicle. So a means to transfer infection, but it itself is not infectious in any way. It's just I put my infection on it, right? So that's indirect. We have direct and we have vector transmission. That's for all different kinds of horizontal. And then vertical, this is like mother to child. Oh gosh. Okay, Robert Koch, he was the guy that came up with a system to identify these. Oh, we skipped this. Why did it go so far? Why did it do that? That was weird. Anyway, so let's go to this. Never mind. We'll go to Robert Koch in a minute. Vehicles and fomites, we kind of already mentioned those. Oral fecal route, this is a pretty serious one. If we're not washing our hands, this is why we wash our hands. It's the oral fecal route transmission of diseases, still pretty common. Air, of course, you want sometimes if you're in a hospital to filter the air because of how many microbes can be transmitted in the air and can hang out in the air for quite a long time as droplet nuclei and aerosols. So dried microscopic residue versus actually suspended from the actively coughing at that time. Yeah, and it depends on the microbe too. So the sneeze and the microbe. So something like I thought I read, and I might be wrong about this, but I really thought that I'd read in the past that like tuberculosis can be in the air if you're coughing, can hang out in the air for like 8 hours or something like that. But it's unlikely to get it from that. But still, I think they measured it in the air up to 8 hours after somebody who had it had been coughing in the area. Anyways, now we're actually at this. Robert Koch, he came up with these postulates to identify these the causes of infection, basically. Okay, so that was that. I can't get into chapter 14. I just don't have the time for it. I really thought we were going to get both of them. I don't understand sometimes what happens with the time. I don't feel like I talk more in this than I do that other class. All right, well, whatever. We'll go over chapter 14 briefly on presentation day. Okay, so there won't be a lot to cover. It's terms mostly.